It's my pleasure to introduce our next speaker, Professor Adam Della Zerda, coming from the Departments of Structural Biology and Electrical Engineering at the School of Medicine at Stanford University. He's also the founder of Visby Medical, and today he'll be talking to us about some of the technologies that he's been working on. So thank you, Adam.
Thanks so much for having me. Let's see, hopefully you guys can hear me well. Okay, so let's see, we click, no. There we go. All right, so before I'm going to tell you what I'm working on, I thought I'll briefly share with you why I'm working on what I'm working on. The why is sometimes more important almost than the what, and rather than giving you some statistics about how many millions of people are affected by this disease or that disease, I'm going to tell you a short story about one person that's very near and dear to my heart, and that's my daughter, Noah. So Noah is 1.5 years old. And my wife and I, first-time parents, are finding ourselves in the emergency room at Stanford. It was really quite scary. She was not breathing well. She was holding up her tummy. She started throwing up, breathed really quickly. Oxygen levels started to drop down. Several ENT surgeons introducing themselves to us, telling us that we really need to know if there's anything in her lungs that's preventing her from breathing well. They try to take a bunch of x-rays of her lungs, but try to tell a 1.5-year-old kid to stand still and not move while she's in pain. That was not possible. They took one x-ray, a second x-ray, a third one. At some point, they're giving up. At that point, they're telling us they wanted to do what's called an exploratory surgery, going into her lungs and just making sure there's nothing there. And they're telling us that if, God forbid, there is something there, maybe a small piece of toy or something that she inevitably inhaled, if it is there, she could develop pneumonia and die within hours. So they're telling us time is of the essence, we need to move right now. They've taken all the measurements they could. They took a bunch of swabs from her, sent it over to the lab, but ultimately, this is what this is. And they're telling us we need to move right now. My wife and I are kind of looking at each other, not quite knowing what to do. We're then basically begging the doctors to do the little as possible that they have to do just to keep her kind of stable until we really know what the heck is happening here. I'm going to spare you the next 24 hours. They hospitalized her. Every 20 minutes, another doctor comes around telling us not telling us, but making us feel that we're bad parents for not letting them operate on our child to do this exploratory surgery, as they call it. It took 24 hours until the result finally came back from the lab. And turns out that result changed everything. The result said that she had RSV, a virus by the name of respiratory syncytial virus. Not a fun virus to have, especially not if you're 1.5-year-old, but also not that bad. There's a way to treat it. Unfortunately, it's already been 24 hours since she started symptoms, so no drug treatment is going to be effective. It's too late. The virus replicated so many times over. It's a little too late for that, but at the very least, we can keep her stable. We know how to approach her, and at that point, nobody told us that we need to perform this exploratory surgery in her lungs. So that was really quite an interesting experience. It took a few days, but eventually her symptoms kind of calmed down and she was better. And we just left with that feeling, wondering, why did we have to go through such bad experience in the hospital? Wasn't there something that we could have done that would have been better? Of course, if the moment we walked into that emergency room, if they could have immediately told us she has RSV, everything would have changed. They would have treated her the right way. They wouldn't take all these x-rays of her lungs. At that age, with a lot of x-rays, that's not fun, not great. Everything would have been much better. In fact, even better than that, if we as parents could have just tested her at home without even going to the emergency room and just go to the local pharmacy, pick up the right prescription, give it to her, and we're all done, that'll be even better. And so that kind of started a process where we started to do some searching and soul searching, really, and asking ourselves, is there a better way to approach this? And so what I thought I'll share with you is kind of what happens the moment you walk into that emergency room. If you start tracking what this swab that they took from her nose, where did that go? Well, it typically ends up in a laboratory that looks like this, typically in the dungeon of a hospital somewhere with a lot of different instruments. One in particular that's relevant for our discussion here is the PCR machine that we all know and love from the last few years for the number of COVID tests that we've been running, many of them on a PCR machine. So those PCR instruments are big, they're expensive, they take forever to run. Because of that, you have to kind of cascade it. So if you have a few samples coming into the lab, you have to wait until you get a few more before you click run and the whole thing starts running. You need to have a PhD in clinical chemistry to even supervise one of these. But the one good thing to say about PCR is that it is darn accurate, both from a sensitivity and specificity standpoint. It is just a very accurate system. Stands for polymerase chain reaction, thanks to Kerry Mullis, who won the Nobel Prize for it. It really is a very good machine. So the question that we've been asking ourselves is, how could we make this whole process be a little bit better? And many times we think about it in terms of speed and accuracy and this and that. I want to introduce another concept. So a month later, after this whole shenanigan stopped, we get the bill from the hospital. Anybody wants to guess how much the bill was for? Okay, 60,000. Okay, these are huge. Just think about it. Just think about what you're saying right now. All she had was RSV. There was a simple treatment for that. She got hospitalized for 1 day and then she left. So let me show you the bill. So you walk into the emergency room. Welcome. That's $11,500 right there. Now they took the first X-ray on her. That was this. Then, remember I told you she kept moving during the X-rays? So that's the second X-ray they had to take after that. Now, on the way to the emergency room, we were actually on the call with the nurse who alerted them to wait for us. They called it a critical case. That's another $1,500 right there, just for the criticality of the emergency visit. That was about 4 hours, because we entered around 8 p.m. We left the emergency room by about 11:30 p.m. They hospitalized her, so spent 1 night in the hospital. So the actual night in the hospital, that's about $7,500. So all in, a little more than $22,000 for that wonderful visit experience. So not good even from that point of view, when we kind of ask ourselves, like, where are we spending our money in terms of healthcare, and why is it that the healthcare industry is $1 trillion and thinking maybe there's some waste somewhere. This is one example. And the reason why I'm telling you this is not because, this is one example that is just very exclusively focused on this one specific example. No, this really happens across the board. When we think about infectious diseases generally, viruses, bacteria, parasitic infections, we humans get infections all over the place. Every place where we have an opening in our body is an opportunity for a pathogen to come in and create an infection. And by and large, not always, but by and large, the way we manage these patients is we treat them first and we diagnose them later. So we treat first, we ask questions later, which makes no sense whatsoever. No other field of medicine behaves like this. We were never going to give somebody chemotherapy before we know for sure they have cancer. You're not going to open up somebody and do an open heart surgery unless you know for sure they have cardiovascular disease. And you're certainly not going to give somebody insulin unless you know that they have diabetes. And yet for infectious diseases, that's exactly what we're doing. It's the only major field of medicine that still works backwards like this. And the reason for that is typically because time is of the essence. And when and where we need the diagnosis, we just don't have it. And so doctors came up with a fancy word to call this, and they're referring to it as giving somebody presumptive treatment, which is just a nice way of saying, I don't really know what you have, so I'm going to guess. And sometimes I'm guessing right, but sometimes I'm guessing wrong. And as you can see, it cuts across many, many different diseases. So the question we then ask ourselves is, how can we take that big PCR machine that is very accurate but not accessible and make it a lot more accessible? And that's a project that we started a few years ago now. It's been a really fun journey, and I'm going to tell a little bit about that journey. The first question we ask ourselves is, what are we shooting for here? And in our mind, we said, the right way to solve this is not by taking the big machine and make it a little smaller machine, because the concept of an instrument and a cartridge would always be very limiting. This is an acute condition. You don't know that you're going to have to test your loved one a week from now for RSV. In fact, you don't even know that they're going to have RSV. Maybe they have something else. It's a respiratory infection. So the idea of an instrument and a cartridge don't quite make a lot of sense for us, especially if we're hoping one day to run those PCR tests in the home. So let's try to preserve the accuracy of PCR, but develop a single-use, disposable, one-time test that is still actually running PCR. If we could somehow take the instrument and the cartridge and merge them together into the same device, well, that's what we're going after. That would be really quite fun. So that's what we've been working on, and fast forward a few years, we did it, and I actually brought this thing to show you. So the device I'm holding here is the world's first and only PCR, single-use PCR machine. And if it's a little hard, oh, you can actually see there we go. It's a little hard to see, that's the whole point, it's really small. But let's go to the next slide here, and you should be able to see it now. There we go, all right, no, that's not the right slide. There we go, that's the right slide. So this is it. So what you're looking at there is an actual single-use disposable PCR machine, and I'm gonna show you how it works and what we did with it. But first, let me just tell you about how it works. It's kind of simple. You see that opening in that top right corner? That's where the sample goes in. This is the first device that we developed. It's for sexually transmitted infections. The sample that goes in there is either a vaginal swab from a female or urine from male. Once you put it in, you just have to close that lid, like so, and it starts running. It's not going to take many hours to run or days. It will give you the result in less than 30 minutes, and you'll be able to see the result by yourself with your own eyes in that panel over there. It can test for a number of infections. Here we're testing for 3, chlamydia, gonorrhea, and trichomonas. Those 3 infections have identical symptoms, but very different antibiotic you need to treat them with. And it really does not tell you when the patient kind of presents based on symptoms. The symptoms don't tell you what's the right way to treat the patient, but this device does. So let me take you through a little bit about how this thing works. And by the way, it's a little hard to do it in this large context, but if you want to come after the talk, I have one of these and we can just open it up and I'll show you what's underneath the hood here. But for now, I'll walk you through the concept. So every PCR machine, whether it's a big instrument or this single-use device, will do 3 steps. We'll be doing sample preparation, where you try to extract out the genetic code from the pathogen. If it's an RNA virus, you want to convert RNA to DNA in a process called reverse transcriptase. Then once you extracted the DNA and hopefully got rid of some of the schmutz, or which is a technical term for all the rest of the thing in the sample that you do not want. Once you finish that sample prep process, you go into the actual PCR reaction where you amplify the DNA, and then you need to detect the presence of the DNA. So this is how we're doing it here. The first step is we're letting the sample go in and get through a heat lysis process where the heat and some of the chemicals that are inside the sample and kind of present underneath the hood over here are going to be acting both chemically and thermally on the pathogen to basically destroy the walls and denature some of the proteins that might be inhibitive here. Once you're done with that, we're ready to go into the next step, which is PCR. But just before we do that, we need to mix in all the reagents for PCR. So you do not need to add any reagents to that sample. It's already waiting for you there. And so that the device has room temperature stability, so you don't have to keep it in the fridge, the DNA polymerase enzyme and everything else that you need, the primers, the nucleotides, the salts, the sugars, are all lyophilized in a small little bead that is waiting for you right here. Oops, sorry. Are waiting for you all over here. So the sample goes in here, we turn on the vibrator, it kind of vibrates a little bit for a few seconds, and then you got your sample there. From there, we're going into the PCR step. The PCR is kind of a pretty cool concept where that takes a fluid back and forth. It's a small microfluidic chamber, and as it's going back and forth like a serpentine, there are actually 3 heaters running across. You can see them right here. There's a central heater and 2 edge heaters. The central heater is set for 95 degrees, so that's a denaturing temperature. The 2 edge heaters are 65 degrees centigrade. So by moving back and forth like this, you're very quickly changing temperature, which is really the basis for PCR. You take the right chemicals, the right reagents with the sample, and you oscillate the temperature. And every time you oscillate the temperature, you multiply the number of DNA by 2. So you do it 40 times, you end up with many, many billions of DNAs, even if the sample initially only had very, very few DNAs to begin with. You can literally amplify a sample that only has as little as 10 bacteria or 10 viruses coming into that port right there. So it's incredibly sensitive from that point of view, but also specific because the PCR reagents are only amplifying what it should amplify, not what it should not. The next step now is to detect the presence of DNA. So we start from maybe 10 bacteria here, we end up with many billions of DNAs, and now we need to detect the presence of it. And the way we do it is, rather than using some fancy electrochemical detection or something, we literally just do an enzyme-linked reaction. So the DNA from PCR floating over and then binding to the surface of the plastic right here. That plastic where it says control spot, chlamydia, gonorrhea, and trichomonas, these are literally single-stranded DNAs that are anchored on the plastic, and the DNA from PCR binds to them or does not if there's no match. And at that point, we flow in enzymes that will then bind and then create a chemical reaction that produces color. Long way of saying it's a colorimetric reaction. If the bacteria is present, you will see a spot lighting up here, and if it's not, you will not. So that's how this works. I thought I'll share with you a little bit about kind of fun stories about this actually being used in the field. What you're seeing here is our one generation ago of how our device used to look. This is a large emergency department network in Phoenix, Arizona. And they used to have their standard of care, patients coming in, presenting with symptoms, and then they will give them the a priori treatment, and then would have to call them again if the result came back different than what they treated. This is again the guessing, not really knowing what you're treating for, and then having to do a correction later on. Sometimes you're lucky enough and the patient actually pick up the phone. Sometimes the patient never picks up the phone, and they're what's called lost to follow-up, which especially in the STD space is incredibly common. And what they're now started doing is they're now using this as a new standard of care, where for the first time patients are coming in less than an hour, they get tested, they get treated, and they know exactly what they have. And if the state allows for that, they even get a partner therapy where they can actually take it and give it to their sexual partner or partners and have them get treated as well for the same STD. I thought I'll share with you one more fun story. That one of the cool things about being able to take those devices out to the field, because it's single use, you don't have to buy a piece of equipment, is that you can really go completely remote. And in this case, it's a clinic that is in the southern part of Arizona. It goes into all sorts of Indian natural preserves, and meeting mostly teenagers that would otherwise not be able to have great access to a clinic. There's one story that I heard from a nurse here that I thought I'll share with you, which is really quite interesting. She told me that when we came and gave them the training on how to use this device and so on, she said, I really think you missed something. You kept telling us that the benefit of a device like this is to find whoever is positive and then treat them for what they have. She goes, I think you're missing out the value of the negative. I said, what do you mean? She goes, look, let me tell you about this patient I had here last week. She came in here and she was worried that her husband is cheating on her, and that's why she's having maybe an STD. I was able in that same visit to tell her that she's clean, referring to the fact that she has no STDs in that case. She said the patient burst into tears, showed her a picture of her kids and said, you just saved this family. And she said, don't underestimate the value of telling somebody that they're negative. So stop just focusing on the fact that you want to detect positives and treat the positives. There's value to being able to detect a patient, tell a patient that they're negative as well, which was a good learning opportunity for me. I'm going to go a little faster over the next couple of slides, but we've run a lot of clinical trials through that. Obviously, you need to do that in order to get FDA clearance or authorization or approval. And so I thought I'll share with you that through a fairly large clinical study that was sponsored by the NIH, we were able to show across almost 2,000 patients that this small PCR machine is just as accurate as a large PCR machine. But maybe much more interestingly, I wanted to share with you is we've done this clinical economic study. So what's that? The idea is not just to prove that it's accurate, which is the only criteria FDA cares about, safe and effective, but we also wanted to understand what is the economical impact on a clinic running such a device. Because ultimately, and it feels horrible to think about it, but financial incentives drive behavior. Period. End of story. It's also wonderful if along the way the technology is helpful to patients, but if it does not make sense economically to a clinic, it will not get adopted. And that's a much longer story that I'm happy to share with you at some point, but I'll share with you this one study that we've done. We went into an urgent care center and we asked them to measure every possible thing that we can measure. To measure anything that has a currency of dollars, minutes, satisfaction levels, or even accuracy of prescription. And what we've been able to show is comparing kind of two groups head-to-head, the standard of care, which is collecting a sample, sending it to the lab, and hoping that we're treating, that presumptive treatment is accurate, comparing that to what you could do if you have a device giving you the result immediately, what will be the impact. So on the time to result, on average, what they've measured is it takes about 2 attempts until you get a hold of a patient to communicate the result to them. The total amount of time being spent just on calling is a few minutes, and the average time between the patient coming to the clinic and actually giving them the result is about 7 days. But you see that this is the median. Hopefully in medicine, we're going to try to do a bit, we care not just about the median patient, we care about maybe the majority of our patients. And you can see that the tail is very, very long, going all the way up to 19 days or so. But in terms of accuracy of prescription, I thought that was really quite fascinating. We just measured what is the rate of overtreatment and undertreatment. Because remember, there are 3 different infections here, chlamydia, gonorrhea, and trichomonas. They all require different antibiotics. And so let's say the patient has chlamydia, but we actually treated her for gonorrhea. What's going to end up happening here is this patient was both overtreated and undertreated at the same time. Now, sometimes doctors know that there's a good chance that this patient will not come back, and so what they do is they actually give all 3 antibiotics to this poor patient all at the same time. Just imagine the consequence of taking that much antibiotic all at once, the impact it has on our microbiota and so on. And you can see here just some of the anecdotal examples of what kind of impact a device like this could have if we're able to give it a result immediately. And within 30 minutes, the patient is still around. We can really reduce dramatically both the over and the under prescription rates. So I thought I'll share with you another kind of fun story that it took us a very long time to assemble this device. We literally worked there assembling and taking about an hour and a half worth of somebody's time to assemble this, kind of like an artisanal diagnostic test. And I'm really excited to share that it took us 4.5 years, but we finally finished developing a fully automated manufacturing line that can now produce this. We got incredible support from the NIH through a RADx project. And because of that, we've been able to move from taking an hour and a half to assemble a single device and bring it down to the point where this automated line can produce a device like this every 5 seconds. So the capability now to really reach a much larger number of people, reduce the cost of production, which then allows this test to be much more affordable at the end, is just so much greater now because of that. It's been a really fun journey. Now, I'll just share with you that this really is a platform. We've always looked at it as a platform. The reason why we chose STIs to begin with is because it felt to us that it's a really challenging problem. It's not seasonal, so unlike maybe respiratory infections that are certainly seasonal. But since then, we've tried to work on a few more devices, and I thought I'll share with you one more here. So this is our respiratory test that we just got through the FDA a few months ago. This one is looking for influenza A, B, and COVID all at the same time. Next year, fingers crossed, we're adding RSV to that, making a full circle. That's what my daughter really needed a few years ago. But it's working really quite well. It's quite accurate, as you kind of imagine. And I thought I'll share this with you. This is kind of a fun slide. We've been planning this vacation for a very, very long time, the whole family. And finally, we're all meeting together for this vacation of ours. And then my wife and one of my kids are not feeling well. And so we ran the test. We were assuming they probably had the same thing because they got sick at the same time. Turns out my wife at the top there had COVID, but my son in the bottom had influenza A. Not otherwise knowing it, we would have put them in the same room together to quarantine. But now that we knew, in less than 3 hours of them showing symptoms, each of them got the right treatment. The one at the top got Paxlovid, my wife got Paxlovid, my son got Tamiflu. And the cool thing about viruses is that if you can detect it early enough, fast enough, and you treat it quickly enough, you can actually stop the replication cycle of the virus. So it makes a huge difference if you start giving treatment a few hours after the diagnosis or after the symptoms or days after the symptoms. If it takes you days, you might as well not bother treating them at all. So I think with that, we'll do a pause here and open up for questions. And just thank you so much for your attention and being here.
Right. Looks like we have one question.
Yes. Given that our medical field is really driven, the payment to our medical field is really driven by diagnostics. So oftentimes a primary care provider or a hospital is more highly reimbursed for diagnosing an illness than actually treating it. So is there resistance from places that are relying on the money they get by sending out for diagnostics?
It's a fabulous question, and you're absolutely right in this. The incentives for running a diagnostic test versus treating are massively, financial incentives drive behavior. So let me just share with you, let's just take urgent care centers as one example. Urgent care centers in the United States are split by and large to 2 groups. Those that are on a fee-for-service basis, That's kind of a pay-per-click. I run this test on you, I charge insurance for that. I do this diagnosis, I get charged a little bit more. Oh, I also run a pregnancy test, I get billed for that as well. And the other group are the capitated system. I'm getting paid to take care of you. Fixed number of dollars per month, that's it. I do more, I do less, I run this diagnostic, that diagnostic, I just imagine what the diagnosis must be and I treat you off of that, and I get paid the same amount. You can see how both of them could have side effects, right? And could be taken advantage of. Much easier to try to introduce a new diagnostic test if it's a fee-for-service basis, where there's an existing CPT reimbursement code. As a doctor, I'm looking at it and saying, hey, instead of the lab making money, how about I do this? I make this money on running this test, right? It's an increased revenue to my clinic. On the flip side, if it's a capitated model, not a fee-for-service, but a capitated model, you don't have that. So all of a sudden you're thinking twice, should I run this diagnostic? Is there maybe a cheaper one? How about maybe I just treat the patient, we'll see. Worst case, I give him some extra antibiotics. No one's gonna die from that, and it'll be okay. So financial incentive drive behavior, that's just the unfortunate reality that we're living in. But as a diagnostic test, we're trying to make a claim for why that's why we're running these clinical economic studies, that regardless of whichever model you're under, we can show that there's real value for you, either as an increased revenue to the clinic, which drives their behavior to run this, which happens to align nicely with what's really in the best interest of the patient, or in a capitated system where we show, look, if you do not diagnose them properly and they go and develop these downstream effects, that'll be really bad, and you as an insurance company will have to pay a lot more later.
Hi, I guess my question kind of the same line. So I just want to see, is it available to buy? It's just in a market right now, the price-wise, and how you want to exactly kind of the question compete with the case that like people go and they just have the test done like in any facility.
Yeah, so these two products that I showed now are on the market. The FDA that we've received is only for point-of-care use, so it could be used by hospitals, urgent care centers, pharmacies, not yet for at-home over-the-counter use. That's something we're working on right now. We're actually running the clinical trial at this moment. So hopefully, fingers crossed, we'll be able to get soon and have an approval for at-home use as well. In terms of price, we're trying to keep it under the reimbursement. Certainly moving to automated assembly really helps to try to keep kind of prices low as possible. The goal is one day to bring it down to a few tens of dollars, but it's still a PCR machine, so it will never be in the same price range as those rapid antigen tests. But on the flip side, you're getting the real accuracy of a PCR, which means you can really count on the result.
Thank you much for the great talk. My name is Ken Golub from the Albert Einstein Hospital in Sao Paulo. And in Brazil, we have, and other places in the Southern Hemisphere, a serious problem with Zika, chikungunya, and dengue. Blood-borne diagnostics, typically, not blood-borne diagnostics. You need to diagnose via blood draw. Have you been able to adapt your system for a blood sample, and is that something that you feel would be pretty quick to apply to your platform?
Yeah, it's a great question. So Zika, dengue, and a few other viruses in that category, we've actually have shown that we can work on blood. A few years ago, that's public knowledge, we took this project with the US military that were really, US Navy, that were really interested in hantavirus. I never heard of hantavirus in my life until they approached us with this, but we've actually done this project. It's a hemorrhagic fever, kind of one of the viruses that causes that. And so we developed the capability for that, and along the line, we also had that capability for that. For viruses that are present in the blood, typically a finger prick is sufficient because the levels of the virus is high enough that a single finger prick is possible. There's a mountain to climb to be able to do something like sepsis. That said, right? So I just want to be clear: when we say we can do viruses in the blood, that's not the same as saying we could do bacteria in the blood, because there's several orders of magnitude of limit of detection needs to be gained to be able to get to bacteria in the blood as well. But yes, Zika, all those things can be done. Yeah.
Time for one more question.
That was a wonderful talk. Thank you. Other sample types, like how, in your experience so far or in your plans, what other types of samples? Like, can you take CSF from spinal taps and other types of samples? Is that possible?
Yeah. So I mentioned before the reason why we chose STDs to start with, and particularly females collected vaginal swab to start with, is, believe it or not, this is by far the most difficult sample to deal with for PCR purposes, because so many things could be present in that sample. Once you've shown that you can work with vaginal swabs, you can do just about anything. Because vaginal swab could have a lot of blood, could have things like shampoos, deodorants, and all sorts of other things. Mucus at varying levels, sticky, but bubbly, and so on. So for microfluidics, that's not great to have bubbles, of course. So once you demonstrate you can deal with that, you can deal with many, many other types of samples. So certainly cerebral spinal fluid is a relatively easy sample for us to work with. We can deal with blood, we can deal with all sorts of, think about all the openings of the body. Wounds, for example, you can do a quick swab of a wound and test that as well. So there are a number of options for samples to choose from. Yeah,
okay.
cool.
Let's thank our speaker.
Thank you so much. Thank you.